Okay, I think we can get started now. I'm Rachel Hostetler, and today I'll be joined by Jeremy Miller, and we will be going over the Annotation Comparison Explorer. And as a reminder, this webinar is currently being recorded. It will be posted online to our YouTube channel afterwards. And this webinar is part of a webinar series called Cell Type Taxonomies A to Z. And you can register for any future webinars that you might be interested in. Again, this will be all posted on our YouTube channel, and we are also happy to take questions live during this webinar. You can post them here on Zoom, or if you're watching this from YouTube, you can also post your questions in our community forum, which the link to that forum is posted in our YouTube description. And today's focus of the webinar will be on comparing annotations across taxonomies. And the Allen Institute has created multiple mouse single-cell RNA sequencing taxonomies over the last couple of years. Back in 2018, there was a joint primary visual cortex and anterior lateral motor cortex taxonomy created, which resulted in 133 cell types. A couple years later, in 2021, a taxonomy that was focusing on mouse, marmoset, and human was created, and the mouse primary motor cortex taxonomy had 116 cell types identified. Also in 2021, a separate taxonomy was created looking at the whole isocortex and hippocampus, where 388 cell types were identified. And then last year, the whole mouse brain taxonomy was released, which resulted in 5,022 different cell types. And so all these taxonomies were created in pretty much different areas of the brain, except for the whole brain, which encompassed all of them. And a lot of these taxonomies were given different annotations or different names of the different cell types. And so one question that many researchers might have is, how do these taxonomies relate to each other? How do cell types in the primary visual cortex and anterior lateral motor cortex, how do they relate to the isocortex taxonomy or to the whole mouse brain taxonomy? And one way to answer that question is through this tool that we'll be discussing today called the Annotation Comparison Explorer. And why is this so important? So if we just focus on one type of cell, the parvalbumin inhibitory interneurons in the cortex, we can see that these cells have been studied and annotated in each of these taxonomies. But when you compare across these 4 different taxonomies, we have a different number of cell types, parvalbumin cell types per taxonomy, and they all have different annotations or different names for their cell types as well. And so if you're someone who's been studying parvalbumin cell types and you're comparing your results to a cell type from the 2018 taxonomy, you might wonder, those cell types, what are their equivalent names in the primary motor cortex taxonomy, or the whole isocortex, or the whole brain taxonomy? And so we can actually answer this question, since all of these taxonomies were done with data from the Allen Institute, and some cells from each taxonomy were actually carried over and used in different taxonomies. So some of the cells from the primary motor cortex taxonomy and the primary visual cortex and ALM taxonomy were then carried over into the whole isocortex and hippocampus taxonomy, reclustered, reanalyzed, and then given different cell type names. And then some of those cells, again, were carried over from the whole isocortex taxonomy and used in the whole mouse brain taxonomy. And so because some of these cells were carried over between studies, we can actually follow individual cells' journeys and trace them between the different taxonomies. And we can answer the question, cell type X in taxonomy, the 2018 taxonomy, what is its equivalent name, its cell type name in, a 2021 taxonomy or the 2023 taxonomy? And so that's just one example of what we'll be talking about today, the importance of annotating taxonomies and comparing cell types between different taxonomies. In the overview of today's talk, first, Jeremy will be going over challenges and opportunities with cell type classification. Then we'll be talking about ACE, or the Annotation Comparison Explorer, and how it works. Then we'll talk about some specific use cases for using ACE, and then we'll end with some Q&A. And now I'm going to hand it off to Jeremy.
Thanks, Rachel. I'm going to share my screen now. Can you see the right screen? Great. Yeah, so I'm excited to talk about this today. This is a tool that I've made with some help, and I'll go into what it is in a little while. So starting with some challenges and opportunities in cell type classification. As Rachel mentioned, the Allen Institute has created several foundational atlases characterizing cell types in mouse, and also in human and other mammalian species. Here's an example of 6 of them that are shown on our on this website that we have. They cover different areas of the brain. In humans, some of them focus on health, some focus on disease. And you know, as Rachel mentioned, cell types will often get renamed with additional data and knowledge, and understanding how cell type names and kind of associated knowledge relate between studies is a major challenge. And so this is not just in healthy brain that we have these challenges. There are many groups that have been studying using single cell omics data to try and understand how cell types change in Alzheimer's disease. Here's a gallery of about 10 of them that have come out in the past few years. And as you may imagine, we have the same problem looking at cell types and disease as we see in the normal brain, which is that it's really hard to know how the cell types line up across studies, much less which cellular and molecular changes are replicated. So this is 8 UMaps from 8 of these different studies, where the names of the cell types are shown, and even things at the level of excitatory neurons. This study calls it EX as the abbreviation. This one calls it EXIT. Some of the other ones call it EXC. For the higher resolution ones, you see this, ones that are just numerically ordered. This study uses gene names, and some have higher versus lower resolution. And you can imagine that it is challenging with these different numbers of cell types, different colors used for describing them, different ways of organizing the data, that when one group says, cell type X is increasing in abundance with Alzheimer's disease, and another group says cell types Y is increasing in abundance, it would be hard to tell whether they're talking about the same thing or not. And then there's another related example, which is, let's say you are someone who is doing single cell RNA sequencing in your own lab and have done your own clustering on your data, and you want to know how your results compare with what we've been published at the Allen Institute or with what some other group has published. You use Map My Cells to map your data, and now you have 2 different cell type assignments for all of your cells. You have the ones you've created and the ones you've gotten back from Map My Cells, and you want to know how they relate. What would be a good way of doing that? And so all of these are annotation comparison problems, in this case, all related to cell type classification. But all of these can be addressed with a single interactive tool that I will be talking about shortly. Before jumping into that, I wanted to briefly mention what an annotation is. Really, an annotation is any information about a cell that can be described with words or with numbers. And what I mean by that is, here is an example of 2 cells. Each row represents a cell. These are patch-seq cells where we've collected morphology, electrophysiology, and transcriptomics about the cells. You can see all of the other cells here if you're interested. And there's various pieces of information that we show about them. 2 of them are different ways of classifying the cells. This is the transcriptomic type, defined as cluster. This is the morphoelectric transcriptomic type, or MET type, which uses multiple pieces of information to define the groups. And you may want to know how these relate. And so this is the cell type classification that I've been mentioning, that we've been mentioning just now. Other things are collected about this as well, like what cortical layer it comes from. So you may want to know, well, which cells were collected from these layer 2, 3 versus layer 4. Those are also annotations. Other things you may want to know is, well, we've collected electrophysiology features, and those numeric values you may want to compare for individual cell types. Like how does this, the action potential values relate in different cell types. What isn't an annotation would be the actual image or the 3D reconstruction of the cell, or this image of this waveform. Those are things that are not really in scope here. Another thing that isn't really in scope for annotations are these large gene expression data sets. While they are technically numeric values, these files are too big, and there are better tools for exploring gene expression data sets. ABC Atlas is one of them. If you're interested in gene expression exploration, I think that would be a more appropriate place to start. Here's another tool, the transcriptomic Explorer, where you can visualize kind of multiple genes in rows by their expression in cell types and columns, or a UMAP, which is becoming a popular way of viewing things, where basically each point is a cell and the expression of GAD1 is shown overlaid on the cells in this case. But this is not what we're going to be talking about today. Okay, and just to highlight another point that Rachel made, that this is about linking studies that use different annotations for the same cell. So I'm showing in like little graphics 3 different kind of sets of mouse brain data that we could compare. One is these 2 studies, the mouse VisP plus ALM and the mouse cortex plus hippocampus data, where data was collected using one particular kind of single cell RNA sequencing data called SmartSeq. The same cells were used in this study and were a subset of the cells used in this study. So you can actually compare for this cell, how does its name change between these 2 studies? Separately, the cortex plus hippocampus data set had cells collected using a different single cell RNA-seq method from 10x Genomics, which is the method that was used for the whole brain study. So there's a different set of cells that will let you do this comparison between mouse cortex with hippocampus and the rest of the brain. And then even another method, spatial transcriptomics, MERFISH, where you can, for cells and context, say what the location is of a given cell and what its cell type is. And so this will let you kind of relate the cell types, locations in the brain versus, the cell types with their location in the brain. But what we can't do is make direct comparisons of something about this cell with something about this cell without using some sort of extra steps that I'm not going to talk about in this webinar. Okay, so then kind of with this as a baseline, as a background, what is Annotation Comparison Explorer and how does it work? Annotation Comparison Explorer, or ACE, is a web-based and R-Shiny application for comparing cell types and other annotations. This is the screenshot of what you see when you go to the page. I have a link as well as the QR code if you want to access it. This was a collaboration between the Allen Institute and Columbia University. I made the app with support from Aaron Oster and Basil Katasik, and using some original code developed by Lucas Graybuck. And then the annotation tables that are up here that I'll talk about were created by me, Kyle Travaglini, and Rachel Hostetler at the Allen Institute, and then Tyann Luquez and Vilas Menon at Columbia University. And Lauren Alfiler made the really cool logo up here at the top. Okay, so when you get to the page, what do you do? I'll go into more detail in a bit about what these things are, but basically this is an app that has to be run with 3 sequential steps. First, you use this top panel where you choose the data set for which the annotations will be compared. These can either be pre-selected ones or you can upload your own. Then once you have a data set, you can then filter your data set to only include your annotations or your cells of interest. So if we're looking at the whole mouse brain that has millions of cells, maybe you're only interested in looking at hippocampus and can subset the data to only include, say, neurons for hippocampus or microglia or something like that. And then once you have this set of cells and the data set that you're interested in, then the magic kind of happens down in this bottom panel where there's various visualizations and statistics for doing the actual comparisons. There's also a 4th component on the left, which is basically the help and the feedback. There's an extensive user guide that basically will go into all the stuff that I'm talking about now in more detail. A link to the tutorial, which will likely just be a link to this webinar, but we may end up creating other sort of more focused video tutorials at a later time. I would love feedback. So if you do use this tool and there's anything you like or don't like about it, click this button and it will open up an email to me, or you can go to GitHub and submit an issue there. All of this is based off of a GitHub repository so you can actually download this on your computer and run exactly the same thing that you see on this website if you're someone who would prefer to work that way. Okay, so let's start with a small example about how ACE works. Basically, as I've mentioned, it compares and visualizes annotations for a single set of data, typically cells, with different annotations for those data, typically the assignments or cell type assignments or cell metadata. In this little example, we're going to pretend like there are 8 cells, 8 neurons. Here's the pyramidal neurons that I drew with PowerPoint. And you can imagine, let's say we want to divide them to small versus big. So in this case, the annotations are the word small versus big. The data points are these 8 cells, kind of this annotation one is kind of the group of annotations that were done here. Then we collect a 9th cell and say, okay, well, actually, we want to call these 3 different groups. Let's say these are the small ones, these are average size, and with this new one, these are the big ones. The question now is, how does our original annotations for these 8 cells compare with their new annotations for the 8 cells? For this comparison, we ignore any of the new data because we can only consider ones that are kind of used in all of the annotations. The way ACE works is we convert this information into a table where each row represents an individual cell or data point, and each column represents a different annotation. So this would be annotation 1 are kind of these calls, annotation 2 are these calls. I've added this 0.1 and 0.2 at the end just to disambiguate the names. You don't have to do that. It just can sometimes make it easier to do that. And then once we've done that, we can upload it to the tool, and we get basically different ways of looking at the data. This is a river plot, which I'll talk about in a minute, but basically it's a way of seeing how the names change from one to another. This is kind of a toy example of what a river plot looks like, but I actually could take this exact table and upload it to ACE, which I did, and this is the river plot view you get from ACE. So it doesn't have to be real data. You can put anything you want in there and get these visualizations back. Okay, so now I'm going to walk through the various components of ACE in more detail. As I mentioned, the first step is the data sets and the inputs. ACE includes a predefined tables which compare annotations of the same cells between multiple public and mouse and human taxonomies. And so that's what you can basically select from up here. And then once you select one, you actually get to see a description of what these data sets and their comparisons actually are, in many cases with DOIs of papers if you want to go out and look at it, kind of read more about this. These also then bring up locations on GitHub. All of the data that's included in these tables is publicly available on the GitHub repository, so you can download it and do whatever you want with it also. And then if you'd like, you can actually upload your own tables. To do that for these top components, you switch it to enter your own data, and then you either put in a location to somewhere on the web if you have your data public, or you can upload it and it's not stored or saved anywhere. So it will remain secret to you, but you can still visualize it in the web app. And then, as I mentioned, the data that you're uploading is this table. The first one is the cell information table that I talked about, where the rows are the cells and the columns are the different annotations for that cell. In this case, I'm showing cells that have multiple cell type classifications and some like a sex and the donor information for human middle temporal gyrus. You can also upload a table that has information about the cell types or the annotations, where in this case, the rows correspond to cell types. So like PAC6 would be one of them, or L6CT_2. Here we have EXC, glia, inhibitory, and astrocyte in the cell type column. And then the remaining columns are kind of any information you want about those cells. So kind of where they are in the taxonomy, what study they came from. This direction is their change in Alzheimer's disease, a more detailed description. Some of these have things like marker genes, where they're located in the brain. You can put anything you want in these columns. It doesn't matter. It'll just display it in a location I'll show you a little bit later. In terms of the data sets that we have available right now, we have one on human Alzheimer's disease that I'll talk quite a bit about more. We have several that focus on in human middle temporal gyrus and on kind of translations between the mouse classification studies. And then we have two focused on mouse cortex, which can be used for comparing the transcriptomic types versus the MET types, or looking at electrophysiology properties, like I mentioned, kind of briefly talked about earlier. There's a webinar on PatchSeq if you are interested in learning more about PatchSeq. Okay, so once you've chosen your data set, the next step is to filter cells in the data set. If you skip this step, you just get all of the cells, and in many cases that's useful, and in some cases it's not. In this case, I'm showing an example from this Yang study of which uses a method that enriches for kind of blood cell types. And we want to focus the comparison on these three different cell types they've defined in this study, capillary, venous, and arterial. And so what you just do is you choose the annotation and the various, I guess the annotation group and the various annotations that you want to focus on. And in this case, only 598 of these 26,000 cells are shown. The reason you may want to do this is what I have down at the bottom, where you can see what this river plot would look like if you have all of the data, where you have a lot of things that already been mapped, you have a bunch of different other cell types, and there's these kind of lines going everywhere. But if we're focusing only on these three types, you can see pretty clearly a one-to-one correspondence between these three endothelial names that we have in our CAD data and what they likely represent from this other study. And so this may be useful for in the future if we want to change the names of these cell types in our studies to match what we see in the literature. And then the final component is that there's several ways of visualizing and exploring the data. I'm going to go through what several of these mean. Here's kind of a gallery of different versions of the data. The one I'm not going to talk about is these bee swarm plots, which are similar to violin plots. It's basically showing the data points where you can, for a given category, you can see how they relate to different numbers. And this can be useful for seeing if, say, there's particular cell type has better QC metrics or more of, higher or lower cortical layer, or maybe has higher electrophysiology, or some numeric variable that you want to compare between categories. Okay, so the first primary visualization is this confusion matrix. In a confusion matrix, the rows and columns each represent different annotations. In this case, this horizontal axis represents cell type labels from one study of Alzheimer's disease, Green et al. 2023, and the horizontal axis represents cell types from a different study of Alzheimer's disease, our Seattle Alzheimer's Disease Brain Cell Atlas, or CAD study. And the dots represent kind of all of the cells that were named this in one annotation and named this in the other annotation. And the dots can be built in different ways, but typically bigger dots mean more overlap, and the colors can encode various things. So this is one way of creating a confusion matrix. Here's another way. In this case, this is showing the Jacquard distance. And you can basically adjust the filter so that these dots represent different things while keeping the axes the same. As one sort of concrete example, if we look at this micro-PBM1 and the relationship to macrophages, there's almost a perfect correlation or relationship between these. And so in the future, we may want to consider calling these macrophages instead of micro-PBM1. Another way of looking at the data that I've showed you a few examples of are these river plots. They're also called Sankey diagrams. This lets you view two or more annotation groups. And so in this case, the right two columns represent the two annotations from the previous slide. And this left column represents a different study of Alzheimer's disease, Matthews et al. 2023. And we're again filtering just to look at the microglia. The boxes will show you the count of the number of cells in each of these annotations. And then the lines or the rivers represent the number of cells with a common annotation at both ends. And so in this case, we can again see the relationship between micro-PBM1 and macrophage by following this river from one to the other. You can see in this case, this study did not group down to as many cell types of microglia as we did in this study, and in this study there were many more. Okay, a final way of viewing the data are these scatter plots where you can compare, in this case, only numeric annotations. So in this case, I'm showing the UMAP coordinates for a set of cells from middle temporal gyres. And the dots would represent basically individual cells where the X and Y axis are their values for these numeric annotations. We can color code them by different annotations. In this case, we're color coding the cells based on their CAD supertype. You can also set it to hover and see different kind of pieces of information about that cell. This is showing UMAPs. These XY coordinates could also represent spatial location, for example, for MERFISH data. As I mentioned, there's typically better ways of looking at MERFISH data for gene expression, but for looking at spatial locations or cell types, this tool is one way of doing that. We can also look at electrophysiology features. This is looking at just ET and IT cells from mouse motor cortex, and you can see that the SAG and the AP width can, just between the two of those, distinguish these kinds of cells. Okay, so that's most of the visualizations. There's one more that I'll show in this use case example, but now I want to jump into kind of how you do these use cases. These are going to be pretty similar to the ones that I've talked about before, but I just want to go through again to how you would use this tool for them. The first is aligning cell type names in Alzheimer's disease. So single cell RNA-seq studies of Alzheimer's all report some cell types changing in abundance in AD, but use different names for the cell types. And as we've mentioned, we want to know how we can tell whether the different groups are identifying the same cell type populations. And this was done by combining data and knowledge integration followed by the interactive data visualization. This data and knowledge integration, how this was done is already published from Gabito, Travaglini et al. As part of our CAD project, where basically they took the 10 different studies of Alzheimer's disease, reprocessed all the single nucleus RNA sequencing data, and then integrated into one space so that the cell type assignments from CAD could be given to the cells from all of these other studies. And then what we did is we compiled all of this information that was from these mappings along with the original cluster for these cells and put it into one table. And we then collected the information about the cell types and put it in the second table. Importantly, this included the direction that they reported in the study of the abundance changes with disease. And so what we have now is basically cells from 11 different studies, their relationships to one another and their changes with disease. And we've included this as a table in ACE. And what does this end up looking like? What we can do is focus in using this other visualization on an individual annotation. So in this case, we look at the CAD supertype that corresponds to SST23, and we pull down these other comparison groups, which are the cell type labels from other studies that included neurons in them. And we can see what those cells were called in, what the SST23 cells were called in those other studies. And so what we find is that for the SST23 subgroup, which is the leftmost one here, 5 of these 8 studies that are color-coded in blue also found that these cells that were in the specific SST type were decreasing in abundance in Alzheimer's disease. So basically, SST interneurons are selectively vulnerable in Alzheimer's disease across multiple studies, which can really improve our ability to believe this result since it's so reproducible across studies. I'm just showing one example, but this is something that you could do for any cell type you're interested in this Alzheimer's disease study. Another example is one that I mentioned before. If you have created your own single nucleus RNA sequencing data set and clustered your data and want to see how your results would compare with cell types on Allen BrainMap. And the way you would do this is by running Map My Cells, followed by visualization in ACE. If you want to know how to use Map My Cells, I would encourage you to watch the webinar, but essentially you upload your data and then you download the cell type assignments. Once you've done that, you can then create a table that includes your cells, the cell type assignments from Map My Cells and your original cluster calls, and then can upload the data to ACE and can use any of these visualizations to show your results. As an example, what I did is I took data from our original study of MTG, this Hodge et al. 2018, used Map My Cells to get the CAD cell type assignments from them. And then I'm showing the confusion matrix of those results. The horizontal axis here is the CAD mapped results. The vertical axis here is the original cluster assignments from that paper. And you can see lots of sort of dark dots along the diagonal and not much else on the side, which shows really good correspondence between the mapping and clustering results. This is looking at all the data, but you may want to jump in and explore subsets of the data. For example, this area with the superficial excitatory neurons is less well in agreement between the clustering and mapping, and it might be interesting to see why that's the case. And then as a final example for comparing taxonomy versions, we've talked about this one quite a bit, that the Allen Institute has extended cell types to cover the mouse whole brain. And we want to know, like, what is your favorite cell type from the study of mouse cortex from hippocampus, now called? And the way you would kind of do this is just look it up in one of the provided tables. I've mentioned the various tables that are currently available. One of them is this comparison of cortex plus hippocampus to whole mouse brain. So you basically choose that data set and then go to the individual cell type, the explore individual annotations panel. I've selected a particular supertype from the cortex hippocampus one called L6IT CTX OSR1. And we can see by choosing the various whole brain cluster or the whole brain assignments as the other metadata we show, what this cell type is now called in the whole mouse brain. I think the most obvious thing is what was originally one cell type has now been split into multiple cell types. If we want to see what those represent, you can basically click on the name of this. And this is the other place. This is the place where the metadata information table really comes in, which is when you click on this, you can see down below information that was about that cell type. In this case, this particular one is only found in isocortex. The name was originally CTX, suggesting it was a cortex cell type. So this tracks. It's in a lot of different cortical areas, so it's not specific to one cortical area. You can see the marker genes for this particular cell type. Another way that we can sanity check this is that these were originally called L6IT something. They're now still called L6IT something. So even though it's been split up, a lot of these are still in the same kind of cell. So these are the kinds of things that you can do to compare between what something was originally called and what it is called right now. So I've only focused on a few use cases, but there are a lot of other places where this is useful. ACE can really be used in any situation where annotation comparisons are needed. One example is identifying which cell types are in which brain regions. I think I briefly touched on that before with the Merfish data. We can also use this to assess single cell omics data quality. So let's say you've done your mapping with Map My Cells, and in addition to uploading your cluster results and your mapping results, you also wanted to upload your quality control metrics, like the number of genes detected or the percent of mitochondrial reads. You can visualize, what fraction or the distribution of those QC metrics per cell type to see if maybe some of your cell types might be outlier cells that you want to remove. Other things that you may want to do is compare changes in kind of gene abundance by sex or disease state. Maybe you have a hypothesis that a particular kind of cell is only found in female or is more predominant in female. You could look that up if you included that information in the in your file. You could also look at abundance changes with disease directly by including that information about the donors in the uploaded information as well. And this doesn't even necessarily have to have anything to do with neuroscience. You could ask questions like, where, what are the political parties of individuals based on their city, state, or like geographic feature if they're in rural versus urban environments, or comparing the age of workers in different professions. I would imagine like preschoolers versus professional wrestlers versus, presidents would have very different age distributions. I presented a few other potential use cases in the user guide, and if you do end up using ACE, I would love to hear other ways that you use it that could help me make this tool more valuable to more people. So overall, just to conclude that ACE is an interactive, user-friendly tool that allows comparison of 2 or more annotations of the same cells. It includes data for translating cell type assignments and knowledge across several published studies in healthy and in Alzheimer's disease brain. And ACE allows users to upload their own tables, for example, to compare clustering and mapping results. And there are multiple options for filtering and visualization to allow a user to ask focused questions. So that's all I have. I'm going to stop sharing, and if there are any questions, I'm happy to take them right now. If you think of a question later, Rachel can remind you of how to pass that information along.
Yes, we have our community forum, which is community.brain-map.org. I'll also post a link to that in the next slide or two. And Jeremy is very active answering lots of questions on the community forum. So if you have any questions for him, you can always put them over there.
I do see one question.
Okay, I see one now. How did you account for variations in methods, donors, and sex in the AD studies?
The short answer is that we didn't account for them. There were in the model that was used for data integration, there were like batch variables that were used to help to sort of align the data in the integration. But we basically took whatever was available. So I guess batch correction is the short answer. But we did take whatever was available, and I think a major source of variation was the different computational pipelines that were used for analysis of the data. And so Kyle spent a lot of time carefully QCing and integrating the data using the same computational pipeline, which got rid of many sources of variation. And then the rest of it was through kind of parameter adjustments in the models.
And the details behind that data integration is in the published paper, correct? That's in the methods section. Yeah, so that was just published in Nature Neuroscience, I believe in November. Gabito and Travaglini are the first authors on that paper, and there's a whole detailed section explaining that integration between the AD studies in more detail. Another question that I thought of is, there's also patch-seq data in the Annotation Comparison Explorer as well, right? Because that I don't believe that was mentioned this webinar. I guess if you could give a few comments or thoughts on that, or maybe potential use cases.
Yeah, so I think there are a lot of valuable tools for exploring gene expression data, as I've mentioned. Maybe fewer of them for patch-seq, but patch-seq, when you the feature set in patch-seq data for the morphology and electrophysiology, as well as the number of cells, is typically dramatically smaller than kind of either axis of either of those for gene expression data. And so just using this as a way of comparing electrophysiology or morphology features for different cell types is something that would work really well with the scatter plot tools or with the bee swarm plot that I briefly mentioned. I don't know if this is necessarily the best way of doing it, but it's a really easy way of doing it. You would just upload basically any CSV file that has that information in it and can use this tool for exploration of the data.
Great. And then I have a quick feedback survey that's just 1 or 2 questions that I'm going to launch. That would be very useful for the participants to fill out. This helps us plan our future webinars and helps us improve for those in the future. And this information is not shared publicly anywhere. The survey results are anonymous. It's just useful for us to collect information on how to make these webinars more useful.
I do see that there's another question.
Oh, great. When you have one annotation mapped to multiple levels of annotation in a different study, how do you resolve the one-to-many mapping scenario?
That is a very good question. I think we avoid that question with this tool. Basically, this will allow ACE will allow you to see that these one-to-many relationships exist, and to in particular in the confusion matrix, it'll often show up as either a horizontal line of dots or a vertical line of dots, where whatever study has the many is where you would see the kind of multiple dots in a row, and the ones that has the one is where you'd see the one. In some cases, you have few to few, you have many to many, one to one. Any permutation of those can show up. I think, I mean, the short version is that cell types are likely continuous space, and we are cutting them up in different ways. And just depending upon how you do it, you're going to get these kinds of mappings. And I think just having a way of visualizing and seeing what these relationships look like is a useful first step. I don't really think I would like to say more than that about it in this webinar, but this is a very important question, like how one would deal with these.
Yeah, I personally like using the river plot function to see that, because for me, I think that's another great way to visualize. You have one taxonomy on one hand, the other taxonomy on the other, and some of them, depending on which level of the taxonomy you're looking at, if you're looking at the subclass level versus, the cluster level, subclass tends to be more consistent, but sometimes, those individual clusters then split into other clusters in another taxonomy, and you can see that pretty clearly in the river plot. All right, thank you everyone for participating in our poll. And then we just have, let's see, do we have, okay. We do not have another question. Let's see, we can hop on back to our last couple of slides then. I can share those. So here, if we go to, as I mentioned previously, this webinar is part of a series of webinars. So if you'd like to hear more detail, particularly about the Alzheimer's taxonomy or CAD, on February 12th, we'll have all of the lead scientists on that study joined together for a webinar on February 12th, going over more details of the study. And then we have some other webinars that might be useful if you're interested in looking more into the differences of single cell versus single nucleus, and differences in those techniques and when to use which. We will have a webinar focused on that on January 22nd. And you can register for all of those at alleninstitute.org/events, or click on our QR code. And again, these are all live, but then they're also recorded and put on our YouTube channel afterwards. And we also have a few other resources here. We have a cell type supportive resource catalog, which is labeled here, brain-map.org, help in community guide cell types. And this catalog has a number of webinars, tutorials, use cases, other documentation to help understand a lot of our cell type taxonomies and tools. And then again, our community forum, which I've mentioned, community.brain-map.org, where Perhaps another person has already asked a question that you have and it's already been answered, maybe. So you can search through community forum questions, or you can also, ask whatever questions you have. And then Jeremy and some of our other scientific staff, and sometimes myself, will go and answer those questions to the best of our abilities. So these are some other ways. If you have, questions following up, especially if you're using ACE on your own after this webinar, definitely post something in our forum so that we can identify your problems and try to provide any assistance. And with that, we'd like to say thank you to all of our funding sources, acknowledgements for everyone involved in creating the tool ACE, those involved here at the Allen Institute and also at Columbia University, the CAD project. Here's a link to the app itself down here. It's a Shiny app, so c-ad.shinyapps.io/ACE app. This is the actual link that will take you to the Annotation Comparison Explorer. And we can also post this link in our YouTube description as well so that it's easier to find. All right, well, thank you everyone for joining us today, and I hope you enjoyed the webinar.
Yes, agreed. Thank you.